The effects of temperature and precipitation on the phenological stages of Acer macrophyllum, or also known as the bigleaf maple, by Marcel, Logan Gray, and Haoye Zhang. For our abstract, global warming is shifting seasonal cycles and influencing plant phenology. The bigleaf maple is a native species with ecological and culturally significance in the Pacific Northwest. We use data from 2009 to 2020 from the USA National Phenology Network, or the USA NPN, and we examined the relationship between spring's minimum maximum temperatures, precipitation, and phenophases onset dates. Our three sub-hypotheses were tested. Higher spring temperatures advance the leaves phenophase, increased precipitation delays the young leaves phenophase, and higher fall temperatures delay the colored leaves phenophase. Scatter plots and simple linear regression indicated that warmer temperatures generally advance leaf emergence in the spring and delay coloration in the fall, while precipitation has had a positive but regional variable effect on young leaf timing. These results enhance understanding of climate impacts on temperature Dulucis trees and can guide future forest strategies. Global temperatures are slowly rising, causing seasonal cycles to alter gradually. The plant phenology is also being affected by climate, and this change is significant in western Washington. The bigleaf maple is a large, ecological, significant broadleaf tree native to west coast of North America. It goes from southern Alaska to northern California. It can grow between 30 and 48 meters tall, and has trunks exceeding 1.5 meters in diameter. The leaves of bigleaf maple are also 30 centimeters wide. Its rough bark is helpful for supporting dense layers of moss and liverworts. It also provides habitats for insects, amphibians, and birds. The broad canopy shades underneath plants, and also the leaves give the soil richness and keep them healthy. The wood is also used to make furniture and musical instruments, and its sap can be tapped for syrup, as well as the trees play a cultural significance to many indigenous communities. In the context of a warming climate, understanding how this species' phenology shifts over time is important for predicting ecological change and managing carbon forests in the Pacific Northwest. Many previous studies have shown that trees are sensitive to temperature changes, and phenological events such as bud bursts, leaf sprouts, and flowering tends to occur earlier when spring temperatures are higher. For example, in eastern North America, parts of Europe, leaf out can advance 2 to 4 days for every 1 degree Celsius increase in spring temperature. However, most of these studies tend to check for global scale trends, or economically viable tree species. The gap in research for bigleaf maples is that the fact that there's no studies within the Web of Science database directly comparing the effects of temperature and precipitation for the bigleaf maple. For bigleaf maples, the leaves phenophase mark the initial emergence of foliage, followed by the young leaves stages. As the leaves continue to develop, the leaf folding stage when leaves reach full expansion. Leaf folding is a fast growth stage that usually happens around 7 to 10 days after the leaves fully emerge. During this time, both the petiole and the leaf blade cells stretch out, allowing the leaf to expand to its full size. The two environmental signals mainly control this process is winter chilling, which helps the tree break dormancy, and spring warming, which promotes cell growth. If winter chilling is too warm, even warm spring temperatures might not lead to normal development, potentially explaining the inconsistencies in patterns for phenological changes. To address this research gap, the study asks, how does spring and fall temperature and precipitation affect the timing of different phenological stages of the bigleaf maple? Using the NPN from 2009 to 2025, we analyzed the relationship between climatic variables and the timing of leaves, young leaves, and leaf coloration phenophases. We hypothesized that warmer spring temperatures advance the leaves phenophase, and that higher precipitation delays the young leaves phenophase, while warmer fall temperatures delay the onset of leaf coloration. By testing these sub-hypotheses, we aim to better understand how multiple climate factors interact to influence plant development. This knowledge can support forest managers, park staff, and ecological researchers in adapting conservation and outreach activities to future climate conditions.
Our study focuses on the Pacific Northwest, Washington, Oregon, and California state. This region has a temperate climate with mild, wet winters and warm, dry summers, making it an ideal place to observe seasonal change. We chose the bigleaf maple, which is a dominant deciduous tree here. We specifically examined three phenophases: the first leaves emerging, the stage of young leaves developing, and later in the year, leaf color in fall. For our data, we used the USA National Phenology Network to obtain it. We look at observation from 2009 to 2020 for spring, and from 2009 to 2025 for fall. The climate variable we focused on were spring minimum and maximum temperature, fall minimum and maximum temperature, and the total spring precipitation. We analyzed the data using the USA NPM visualization tool. To reduce uncertainty, we applied precision filters, 14 days for spring data and 30 days for fall. We then used scatter plots and simple linear regression to test our hypothesis. In each case, the dependent variable was the onset data of the phenophase, measured as the day of the year. The independent variable were temperature and precipitation. For the result, these two are the scatter plot between spring temperature and the onset day. For minimum spring temperature, values range from about 4 to 11 Celsius degree. Leaves onset day range from around day 70 to 115 of the year. There is a negative trend. Higher temperature correspond to earlier leaves phenophase. The R-square was 0.4, showing a moderate relationship. For maximum spring temperature, values range from about 12 to 20 Celsius degree. The day is between 70 and 115. The relationship are stronger. The R-square is 0.53, meaning daytime temperature has greater influence on leaf emergence. Next, spring precipitation and the timing of young leaves. In Washington, spring precipitation ranged from 0 to about 500 mm, and the young leaf onset occurred between day 19 and 113. The relationship was weakly positive, with an R-square of 0.24. In Oregon, precipitation ranged from 0 to 418, and onset day were between day 19 and 114. This showed a much stronger positive correlation, with an R-square of 0.76, suggesting rainfall plays a more important role in Oregon. Finally, fall temperature and leaf coloration. For maximum fall temperature, values range from about 12 to 20 degrees, and leaf coloration was observed between day 243 and 333 of the year. Here we found a positive relationship. Higher temperature delayed leaf colored, with an R-square of 0.43, which is significant. For minimum fall temperature, the range was about 4 to 11 Celsius degree, with coloration again between day 243 and 334. There was a slight positive effect, but R-square was only 0.15, which is not meaningful to us.
Our results. Results for the temperature analysis for spring clearly showed a negative relationship between the spring temperatures and the onset of leaf phenophase for the bigleaf maple, with the minimum temperatures having correlation of 0.4 supporting that individual hypothesis, with the higher maximum temperatures having an even greater negative correlation at 0.53, also supporting the hypothesis for Washington, Oregon, and California data. For the precipitation values for spring for Washington, had relatively low but still significant correlation to support the hypothesis for the effects on young leaves, while for the Oregon dataset, it had an even greater correlation at 0.74. The precipitation dataset had one of the highest of any of the R values. While for fall, bigleaf maples that encountered a higher max temperature had a positive correlation with delaying the time of onset, supporting the hypothesis, with a R value of 0.43 for the range of Washington, Oregon, and California, while the min values had a positive of 0.15, though it was positive, it did not have enough significance, so the result is inconclusive. Limitations to the study was the relatively low number of observations. 14 observations for the spring minimum and maximums, and 9 for the minimum and maximum for fall. For precipitation, Washington had 7 and Oregon had 13. General lack of another limitation was the general lack of data entries, with 2018 and 2020 for spring being the most relevant, most recent. Both the limitation of number of entries and the range of data that we have to work with disallowed comparison between different temporal states for each of the states, along with just providing better linear regression formulas, as the lack of data do allow for a more inaccurate number. The future studies recommendations for those performing them is to extend the time frame by another 10 years to include a broader range of temporal data, along with the expansion of recorded data entries to better consolidate individual regions, both Oregon, Washington, and California. They should also focus on looking at how the changes in the current phenophase timings are affecting other processes such as pollination, species competition, and carbon cycling, as those are not the focus of this study. We'd also like to thank Dr. Yan-Chu Wang, along with the USA National Phenology Network, for providing the data with the research, along with the tools to visualize it. Data was once again provided by the US National Phenology Network, along with the participants who collected the data for their data set.